politics. But I think it's really incredible for those of us engaged in stem cell research or examining the ethics or societal issues involved that these things have walked in tandem. Stem cell science has been very much a public and societal issue from the new age of stem cell science, and I think we need to address some of these issues today. The panel we have together is fantastic, and what the panel is going to do is just sketch some of the familiar issues quickly in ethics and societal issues related to stem cell research, and then move us forward. What's the next set of ethical issues and the types of scholarship that we need to sort out with where the science is going and how we're going to be responsive to it. I'm going to speak, and then I'll introduce the other panelists, and then they'll each speak, and if time permits, we'll have some time for question and answers. So it's no news to people in this room that scientific excitement for stem cell research of all sorts is extraordinarily high. And part of this is driven by the desire for access to investigational treatments, especially for devastating disorders. Think of the advocacy, think of the rhetoric, think of all of the attention that's been focused on embryonic stem cell research and its progress because of this great excitement. As you've just heard, terms in the millions and the billions, the amount of money spent on this research and much life science research is incredible, and yet we also know that the moral stakes are extraordinarily high. I'll pick an example, not from Maryland, to start. For those of you who don't know, what you see here is a picture of Woo-Suk Wang, the supposed first discoverer of being able to do somatic cell nuclear transfer. He's very excited in that picture, and the reason why is he got to be named by the Korean government as the supreme scientist. Now, everybody up here has got named chairs at universities, but a supreme scientist is very cool. Everyone would want to be called the supreme scientist. What you saw was Korea invest an extraordinary amount of money into the idea that they could make Korea the hub for certain aspects of stem cell research, and they issued a stamp, which you see on the lower portion of that slide. And if you look at the left side of the stamp, you see what is a depiction of SCNT, or somatic cell nuclear transfer. You see some bright green neon going across that. I'm not sure what the green neon is, but it's supposedly an injection to this person who's in a wheelchair. And you see the person in the wheelchair get the stem cell injection, begins to stand up out of his wheelchair, begins to walk and then jump and then make new stem cells. Now, what you see in the next slide here, though, is shame. And this is shame when it was discovered that this was fraud. Was this a failure of science? It's probably no. Science caught it. You cheat, you lie, you steal, you get caught. That should be the lesson. What happened was it sort of undermined our hopes, but the reality of science was we couldn't quite do things the way they had been reported. It just wasn't that easy. Now, many of the debates that we've had around the globe, not a surprise, related to the embryo. The moral status of the embryo is debated. Long-standing things about whether it's morally acceptable to destroy embryos to create human embryonic stem cells, or is there a distinction between using discarded embryos versus those created for research? These debates were long and hard and difficult and sometimes intractable. Professor Haimes will talk later today about additional approaches to getting around, if you will, some of these issues and her social scientific studies of that area. We also had debates about ethics and oocyte retrieval. It's not easy to get human eggs. Hormonal treatments are used to treat women, have side effects. They are very real. It is very arduous. And what are the effects of paying or not paying women? A set of debates that have been rekindled by different state efforts in this area. We've also heard concerns about chimeras, which Professor Moreno will talk about later. We do make chimeras all the time when we do bone marrow transplantations, but the notion of chimeras in fiction and in the press and when using things across species begin to raise some very difficult issues. Furthermore, the early debates of the ethical and societal issues with respect to stem cell research unearthed a set of cultural and religious practices that were different for many sources of stem cells. Even going back to umbilical cord blood stem cells, there are vastly different cultural traditions between the use and meaning of the placenta and how it's treated at the time of delivery and what it might mean to own something. There are laws, domestic and international, which others on the panel will talk about, where it is illegal in certain jurisdictions to destroy an embryo to create a human embryonic stem cell line. For instance, if a German scientist, Germany has a law like that, comes to Johns Hopkins to do a fellowship and wants to create a human embryonic stem cell line, if she derives a new line and then goes back to Germany, she would be possibly liable for criminal prosecution. Now, what does the lab director do? What does the science editor do with that information? Should that person be working on that kind of science? How do we know about all the differing rules and laws around the globe, domestic and international? And funding issues. As you heard today, we now have a partnership, as of, I guess, the applause outside, with CIRM to encourage collaboration across states. These are very complicated issues, but with each state rules, with each state's funding situation, there are different rules for how they can be used that need to be negotiated. In light of all this, professional guidelines were issued, as everyone knows. The National Research Council and the Institute of Medicine, the National Academies released theirs. Professor Moreno played a major role in getting those going. The International Society for Stem Cell Research also entered guidelines then to look at stem cell research more broadly. Both of these sets of guidelines, the governance models for both sets of guidelines are fairly familiar. They smell a little bit like Institutional Review Board or Research Ethics Review Board, Research Ethics Committee Review, or Institutional Animal Care and Use Committee Review. The notion was somewhat local review by a group of people who are uninterested, not because it's boring, but don't have a personal interest at stake with respect to that research, to look at it to make sure it's ethically appropriate. The escrow committees would therefore be composed of people of various abilities and expertise. But this didn't stop our attention to the ethics of stem cell research and the issues. We have some vexing questions about intellectual property that began with the patenting of some cell lines and thinking forward, what does this mean? What will it mean for the use of either public or private funds to be used in stem cell research at the basic level by the time it comes to clinical translation? And will that improve or impair the ability of people around the globe to have access? Professor Caulfield will be talking about that. Determining provenance. Where did these stem cells come from? Were they ethically derived? Has created a knotty set of questions that have been very difficult. I serve as the co-chair of the escrow at Johns Hopkins, and we face some very difficult issues looking at that. Cell types pose issues, whether it's bone marrow, cord blood, even iPS cells. So the hope of iPS also raises another set of ethical issues, which some of the panelists will discuss. And finally, the area I'm going to focus on is what happens at the stage of clinical translation. This has been the hope of many of those of us engaged in stem cell research or its oversight, is that someday we're going to be using sources of stem cells in unique ways to cure disease. And it's this set of issues that will be the next wave of issues as especially human embryonic stem cell and iPS cells move closer to trials and market that we're going to have to face. Now notice the background conditions here are several. We've been assuming in large part that we're doing translational research, moving things from the bench into animal models and then first in human trials through a staged set of processes that go over time. Yet in other cases, clinical care is being done with sources of stem cells such as bone marrow and cord blood all the time. Can't ignore that, right? So we have a lot to learn from. If you also think that innovation is the way that many things that we do in science and medicine have come to fore, not because of a staged model of translation, but because someone discovered something that was radically different and it worked. Transplantations of different sorts were innovations. They weren't sort of staged processes. Many devices were innovated gradually over time. And so the model that we've been assuming at all times may fail, it may work, we don't know, but we need to be sensitive to these models. What's screwing things up, to use